Hello there, micro students. This is the first lab that we get to try to do online. So the plan is to make some videos to try to capture what you would have missed or what you are going to miss because you're not able to attend lab. So the first lab is designed to better understand a fungus. And so if you recall from long ago in lecture, there are two different types of fungal organisms. There are yeast and molds. So not surprisingly, this lab is focused on understanding what mold and yeast look like at two different levels. So we're going to talk about macroscopic, or what you can see with your eye, and then also microscopic evaluation of fungus. So we're going to look at the learning objectives. And so I already mentioned that microscopic and macroscopic differences are going to be a primary focus. And so at one level, we want to understand how yeast differ from mold, but then also we want to understand how we can differentiate between different mold organisms. And so there are characteristics, again, both on the macroscopic and the microscopic scale that we can use to help us identify and differentiate within molds. So if you recall, molds are multicellular. So they are made up of not a single cell, but rather multiple cells that work together to form one organism. And they differ from yeast in that they typically use asexual spores to reproduce. And just to contrast, most yeast are unicellular and are going to reproduce through the formation of buds through a process called budding. And we'll see that towards the end. So one thing that you all are well aware is that mold has a fuzzy appearance. So whether that's old cheese or old bread, you can tell it's contaminated with mold because you see this fuzziness growing on it. And so the term we use to describe that fuzziness is called mycelium. And so this is what you see with your eye. But when you zoom in and look more closely on a microscopic level at a mold, what you see are these long tube-like structures that are called hyphae. And so the hyphae not only grow up, and that's the fuzziness that you see, and so those are called aerial hyphae, but then there's also these vegetative hyphae. And so those are hyphae that grow into the nutrients. So if you had, say, an orange that has mold growing on it, not only is there the fuzziness on the outside, that would be called the mycelium that you see, you have these hyphae, these tube-like structures that are growing down into the orange peel and into the fruit itself that's extracting nutrients from it. At the ends of aerial hyphae, we have asexual spores. And so what we want to use to identify and differentiate within mold are macroscopic characteristics. And so we can look at how tall are these aerial hyphae. So how big is that fuzziness that we see? And so we call that the height of aerial hyphae. Also, as mold grows, there is a pigment that can be attributed to it. And so as we look at the mycelium or that fuzziness, we can say, well, what color is that? And so we can look at what's called the surface pigment. And then there's also the pigment of that vegetative hyphae. So as it grows down, is there a different pigment? And so we're going to look at two different pigments. We also have the microscopic structure that we can look at. And so we mentioned those tube-like structures are called hyphae. So we can look specifically at the hyphae, and they can have, excuse me, either partitions that are called septa. So those are called septate hyphae, or there can be tubes that lack those partitions. And so those are called aseptate hyphae. But probably most importantly on a microscopic level is looking at how the mold spores are arranged. So this is called spore morphology. That's a term we use frequently to describe the shape or the structure of something. So the spore morphology. So we're going to look at three different mold species, really genus. And so the first is called Aspergillus. And then again, macroscopic is what we're going to evaluate first. So this you can see is mold growing on a plate. And we use a specific type of media to grow mold. And so you read about this in the lab manual. It's called Sabarog dextrose agar. And so on the surface of that agar, you can see this mold growing, and it's very fuzzy. This is the reverse side of the plate, so the bottom view, and you can see the difference in coloration. And so one thing we want to look at is the surface pigment. And so hopefully you can conclude that this particular mold, Aspergillus, has a black color to its pigment, but that same color is not present as you look at this, what we call reverse pigment. And so the surface pigment is black, but the reverse pigment is more of a cream color. The other thing I mentioned, we want to characterize and evaluate the height of the aerial hyphae. And so Aspergillus is said to have medium to high aerial hyphae, and the microbiological term for that is velvety, because it kind of looks like a sheet of velvet there. So it's got some fuzziness. Other molds are going to have less, a lower height of aerial hyphae, but others might have higher. And so that level or that height of aerial hyphae is known as velvety. The next thing we're going to look at with Aspergillus is the microscopic characteristics. So this is what's called a mold slide culture. And so this is a way to evaluate and grow a mold on a slide so that you can then visualize it microscopically. And so under the microscope, what you'll see are these tube-like structures. Those are again called hyphae. And then at the end of hyphae, you can have the development of these asexual spores. And so while all mold are going to have these hyphae, the way in which the spores are organized and how they're formed are going to differ across the different mold organisms. So we can look at spore morphology. So this is under a light microscope. We don't have an electron microscope, but if we did have one and you zoomed in, you could see this arrangement. And so each of these little circles is an asexual spore. This is a nice drawing I found to kind of visualize how those spores are organized around the end of a hyphae. And so one thing that's helpful to do is to think about what this looks like. And so many people have said, and I see this as well, that these spores are kind of organized like the head of a dandelion. And so there's this central location where all the spores are organized and attached to. So when we look at this, we can say, yeah, this is definitely a mold because I can see hyphae and I can see spores. But beyond that, we can also say there's a specific way in which these mold spores are organized. And similar to the head of a dandelion, there's a bunch of spores that are localized around this central area at the end of an aerial hyphae. And so that's what Aspergillus can look like both macroscopically and microscopically. The second mold species we want to look at is Penicillium. That name might sound familiar because this is the species that can and does produce penicillin, the antibiotic that we talked about. So this is again grown on a Sabarad dextrose agar plate. And you can see both the surface and the reverse pigment for this organism as well. And so the surface pigment is more green in this mold species, and the reverse pigment is not green, but rather it's yellow. We can also describe the height of the aerial hyphae, and this organism, this mold species, is not as high. So its aerial hyphae are not as high. And so we call this not velvety, but woolly. And so it's got a slight woolly appearance to it. And so that, again, that is the microbiological term we use to describe it. So what does Penicillium look like under the microscope? So again, we can visualize this by doing a mold slide culture that you read about in the lab manual, and also I produced a video that you can watch for that. So under the microscope, what this mold species look like is, again, these tube-like structures are hyphae, and at the end you can see this spore formation. And so the spore morphology of Penicillium is more branched. So rather than being kind of organized like a dandelion, you have these splitting into 4 or 5 different, they almost look like fingers. And so if you zoom in on it, again, we don't have an electron microscope, but this is what they would look like. So here's again a drawing. And so many people say they kind of look like the wrist and hands of a skeleton. So that's kind of a similar sort of picture. And so that's how you can start to identify this as Penicillium, both macroscopically on a plate. It's got that greenish coloration to it. The height of the aerial hyphae aren't as tall, but then it has a specific spore arrangement, kind of like the hand, a skeleton hand. I didn't use this term, but this is the first introduction to it. So when a mold makes a spore at the end of hyphae and it's not contained with any larger sac-like structure, these are called conidiospores. So both the first organism, Aspergillus, and the second, Penicillium, make spores in the format that we call a conidiospore. And we're going to contrast that with the next mold species that does it a little bit differently. So Rhizopus, this organism doesn't make conidiospores, but it's going to make another form that we'll see momentarily. So again, we have a Sabarod dextrose agar plate, and this organism has a white mycelium, and so you can see how white this surface pigment is, but then also on the reverse side, it's the same color, so it's white in both of those instances. But then also its aerial hyphae are extremely high. The lid has been removed here, and you can see that the mold has basically grown to where that lid was, and if that lid wasn't there, it would continue to grow. So it has a very high aerial hyphae. The term we use to describe this is called cottony. And then microscopically, this is a bread mold, one species of Rhizopus, and this starts to show you how its spores are going to form. So again, you have these hyphae, but what is different about this organism is that there is an actual sac-like structure that the spores are within. So this is not a single spore, but what I'm outlining right now is the edge of a sac, and then inside of this is a bunch of spores. So here's an electron micrograph. It's a little hard to see, but all of those spores are contained within a thin kind of membrane or sac. And of course, these have names. So the spores inside the sac itself is called a sporangium. And so the spores inside of the sac are called sporangiospores. And again, that differs from the two previous mold organisms that had not spores inside of a sac, but rather just at the end of hyphae. So those were called conidiospores. So we want to transition and talk about yeast. So yeast are unicellular, mold are multicellular, and yeast reproduce through the formation of buds through a process called budding, and that differs from what we saw in the mold, which use asexual spores. So yeast are unicellular and reproduce through budding. So on a plate, so a macroscopic appearance, you won't see fuzziness, but you'll see things that look more like bacteria. So these are colonies that you're going to see. And then under the microscope, you don't see tube-like hyphae, but instead you see individual cells, because again, they're unicellular, and they tend to be roughly circular in shape, so we call them globular. And many of those cells will be undergoing the process that we described before of budding, and so that's what we hope to see. So this is what yeast looks like on a plate. Again, we're using a Sabarod dextrose agar plate. And they look just like colonies, and that's indeed what they are. One thing that you may recall is that bacteria also form colonies. When you have a well-isolated colony, you can then say that's derived from a single cell. That's also true for yeast, and so this colony that I'm showing right here is derived from a single yeast cell that's budded over and over again until you have many cells there. And so you have what's called a colony. Yeast colonies are considerably larger than bacterial, and they're nice and perfectly spherical. And they also have a very white coloration to them. You don't get this, but if you were to open this plate, many of you commented about how poorly bacteria smell and that it's never fun to smell E. coli or any of the bacteria. If you were to open this plate, it would smell like you were kneading dough. So much of that smell you get from baking bread is attributed to the yeast. And so this would be like raw dough, which has quite a nice smell to it. So this is just a zoom in view of this colony. So again, see how nice and perfectly round they are. They actually are considerably taller. So the term we use is elevation. We can't see this extremely well from this angle, but they have a higher elevation as well. So we obviously didn't get to do this in lab, but this is a picture I took of a wet mount. And so this is just taking yeast and putting it onto a slide with some methylene blue. And so this is a single yeast cell with a bud coming off of it. Again, there's another yeast cell. So unicellular, that's one characteristic. They're roughly circular, but some of them are a little more elongated. But you can see many of these cells are undergoing budding. So there's one. And this is a prepared slide. So these are the ones that we have in the green slide boxes that we have available for you guys to view. And so again, here is a single cell with a bud coming off of it. So unicellular yeast cells. Very different appearance than what we saw for mold. So no hyphae here, no spores here, but unicellular globular cells with buds. This is again an electron micrograph, and so you can just see a single cell and a bud coming off of it. So just to kind of recap, the major goals of this lab is understanding that molds have a characteristic appearance, but that there are certain characteristics that you can use within a mold when you're evaluating mold to say what mold might that be. So we're just using 3 specific molds, so Aspergillus, Penicillium, and Rhizopus. So both macroscopic and microscopically, can you identify the differences between them? Another way of addressing that is just being able to speak directly to what can we use microscopically across all molds. So rather than just talking about these specific 3, what can you look at microscopically and macroscopically of a mold that can be informative to help you identify what that mold might be? And then lastly, we obviously want to take a step back and understand the differences between yeast and mold. And so looking at both macroscopic again and microscopic characteristics that you can use to say, oh, this has to be a yeast for these reasons, or this is a mold for this reason. So I know that's not exactly the same as doing the lab itself, but that is how we're going to have to do things. This will also give you a taste for how our other labs are going to be. So it'll be a combination of both videos and pictures and me narrating things. So congratulations, you just finished your first lab. Of course, you have an assignment to do and submit through Canvas as well. So make sure you don't forget to do that. Thanks.